Very good morning to all the dignitaries, respected guests, learned delegates from academia and corporate, my dear colleagues and students. I'm Shraddha Tiwari and I feel privileged to welcome you all on behalf of Dayanand Education Societies, Dayanand College of Pharmacy and Dayanand Institute of Pharmacy to the 2 days international e-conference on COVID-19 era, need and challenges in healthcare and pharma sector. A thought we truly believe in is success isn't overnight. It's when every day you get a little better than before, it adds up. We would like to show you a more detailed preview on Dayanand College of Pharmacy. I request our technical team to please share video.
दयानंद शिक्षण संस्था लातूर आजच्या गतिमान स्पर्धेच्या विस्तीर्ण अवकाशात गरुड भरारी घेण्यासाठी विद्यार्थ्यांच्या पंखात गुणवत्तेचं बळ आणि विचारांना विद्वत्तेची धार असावी लागते. 1961 साली असाच गुणवंत, कीर्तिवंत, यशवंत विद्यार्थी घडवण्याच्या उद्देशाने दयानंद शिक्षण संस्थेच्या रूपात लातूरच्या मातीत एक बी रुजवण्यात आलं आणि आज तेच बीज गुणवंत विद्यार्थ्यांनी बहरलेला, विविध विद्याशाखांनी विस्तारलेला असा प्रचंड ज्ञानवृक्ष बनले आहे. आज या ज्ञानवृक्षाच्या छायेत कला, वाणिज्य, विज्ञान, आर्किटेक्चर, इंटिरियर डिझाईन, विधी आणि फार्मसी या विद्याशाखांमधून हजारो विद्यार्थी ज्ञानार्जन करीत आहेत. दयानंद कॉलेज ऑफ फार्मसी ग्रामीण व शहरी दोन्ही भागातील विद्यार्थ्यांना उत्तम दर्जाचे शिक्षण मिळावे या उद्देशाने 2009 साली दयानंद कॉलेज ऑफ फार्मसी सुरू करण्यात आले. क्षेत्रातील आव्हानात्मक गरजा लक्षात घेऊन केवळ आपल्या भागातच नव्हे तर संपूर्ण राज्यात किंबहुना देश पातळीवर उत्तम ठरतील असे फार्मा पदवीधर याचे ध्येय आहे. स्वतंत्ररित्या B.Pharm व B.Pharm हे अभ्यासक्रम चालवले जातात. 22 एकर अशा विस्तीर्ण जागेत विविध सोयी सुविधांनी युक्त अशा परिसरात महाविद्यालय आहे, जे लातूर शहराच्या अगदी मध्यवर्ती ठिकाणी आहे. महाविद्यालयास AICTE, PCI व महाराष्ट्र शासनाची मान्यता प्राप्त आहे. त्यामुळेच विद्यार्थ्यांना विविध शासकीय, खासगी शिष्यवृत्ती प्राप्त होतात. महाविद्यालयातील प्रवेश केंद्रीभूत प्रवेश प्रक्रिया पद्धतीने होतात. फार्मसी या विषयात पदव्युत्तर पदवी तसेच डॉक्टरेट संपादन केलेली तज्ञ मंडळी येथे अध्यापनाचे काम करतात. ICT वर आधारित टीचिंग लर्निंग मॅनेजमेंट सॉफ्टवेअरचा वापर करून अत्याधुनिक तंत्रज्ञानाच्या जोडीने अध्ययन अध्यापन केले जाते. आधुनिक प्रयोगशाळा, अद्ययावत ज्ञान व प्रयोगशील शिक्षण यासाठी सुसज्ज अशा प्रयोगशाळेत विपुल प्रमाणात विविध रसायन व इन्स्ट्रुमेंट्स उपलब्ध आहेत. आधुनिक UV स्पेक्ट्रोफोटोमीटर, इलेक्ट्रोलॅब डिसोल्युशन व ब्रुकफिल्ड डिस्कोमीटर यावर विद्यार्थी निपुणपणे काम करत असतात. स्पर्धात्मक परीक्षेच्या युगात स्पर्धा परीक्षा तसेच राष्ट्रीय स्तरावरील GPAT, CMAT व NIPER यासाठी तज्ञ प्राध्यापकांमार्फत तयारी करून घेतली जाते. याचेच यश म्हणून 50हून अधिक विद्यार्थ्यांनी राष्ट्रीय स्तरावर यश प्राप्त केले आहे. सेमिनार, गेस्ट लेक्चर, वर्कशॉप व इंडस्ट्रियल ट्रेनिंग हे विद्यार्थ्यांना रोजगार अभिमुख बनवते. त्यामुळे गेल्या काही TCS यांसारख्या कंपन्यांमध्ये विद्यार्थ्यांची निवड थेट महाविद्यालयातूनच झाली आहे. मेडिसिनल गार्डन औषध निर्माण क्षेत्रात शिक्षण घेणाऱ्या विद्यार्थ्यांना दिव्य अशा औषधीय वनस्पतींची माहिती व्हावी या हेतूने ही बाग साकारली आहे. महाविद्यालयाचे ग्रंथालय एक सुसज्ज ग्रंथालय आहे, जेथे 7000हून अधिक पुस्तकांचा संग्रह आहे, ज्यात वैशिष्ट्य पूर्ण संदर्भ ग्रंथ, ई-बुक्स, विविध जर्नल्स, राष्ट्रीय व आंतरराष्ट्रीय पातळीवरील ग्रंथालयातील व्यवस्थापन संगणकाधारित आहे. ग्रंथालय हे सकाळी 8 ते संध्याकाळी 8 पर्यंत खुले असते. आमची संगणक प्रयोगशाळा ही सुसज्ज अशी आहे. विद्यार्थ्यांना सर्वांगीण ज्ञान मिळावे याकरिता वर्कशॉप, उद्योजकता, विकासाच्या दृष्टीने विविध उपक्रम आदींचे नियमितपणे आयोजन केले जाते. नियमित अभ्यासक्रमाला शोध निबंधाचे सादरीकरण, पोस्टर प्रेझेंटेशन, इंडस्ट्री व्हिजिट आदींची जोड दिली जाते. शिकून बाहेर पडणाऱ्या विद्यार्थ्यांकरिता विविध प्रतिष्ठित कंपन्यांमधून प्लेसमेंटची सुविधा ही देखील महाविद्यालय पुरवते. विद्यार्थ्यांमध्ये सामाजिक जागरूकता वाढावी या दृष्टिकोनातून राष्ट्रीय सेवा योजनेअंतर्गत रक्तदान शिबिर, वृक्षारोपण कार्यक्रम, स्वच्छता अभियान आदी उपक्रमांचे आयोजन केले जाते. येणाऱ्या विद्यार्थिनींकरिता स्वतंत्र वसतिगृह ही दयानंद महाविद्यालयाची अजून एक उल्लेखनीय बाब. शिक्षणाकरिता घरापासून लांब राहणाऱ्या विद्यार्थिनींना घरच्या मायेची बिलकुल उणीव भासू न देणारं हे वसतिगृह आवश्यक त्या सर्व सोयींनी सुसज्ज आहे. विद्यार्थिनींचे आरोग्य उत्तम राहावे या उद्देशाने इथे पौष्टिक आणि सात्विक आहार दिला जातो. तसेच त्यांची नियमित आरोग्य तपासणी देखील केली जाते. या सगळ्याच्या जोडीने विद्यार्थ्यांचे मानसिक आरोग्य देखील उत्तम राहावे म्हणून त्यांना प्राणायाम, योगासने आदींचे देखील खास प्रशिक्षण दिले जाते. स्वतंत्र अद्ययावत व्यायाम दयानंद शिक्षण संस्थेने केली आहे. जोडीला विविध क्रीडा प्रकारांची प्रशस्त मैदाने देखील आहेत. खेळाडू वृत्तीला चालना मिळावी याकरिता क्रीडा महोत्सव, आत्मविश्वास वाढावा, सभाधीटपणा वाढावा याकरिता विविध चर्चासत्रे, परिसंवाद, प्रेझेंटेशनचे देखील आयोजन केले जाते.
Thanks for watching. We would like to proceed with the inauguration. Now, for inauguration ceremony of international conference, we are having Honorable Shri Lakshmi Ramanji Lahoti, President, Dayanand Education Society, Shri Rameshji Biani, Secretary, Dayanand Education Society, Honorable Shri Ajinkiji Sonone Sir, Management Member, Dayanand Education Society, Honorable Shri Vishalji Lahoti, Management Member, Dayanand Education Society as our guest. We are having Dr. Sureshji Bhattar Sir as our guest of honor from Management Representative. We are having Honorable Dr. Joginder Singh Ji Bisen, Pro Vice Chancellor, Swami Ramanand Tirth Marathwada University, Nanded, as our chief guest. It's an immense pleasure and honor to welcome all the dignitaries. Now I request Dr. K.L. Satpute, Principal, Dayanand College of Pharmacy, Latur, to please deliver welcome address.
Thank you, madam. Very good morning to all. Respected chief guest of today's program, Dr. Jogendra Singh Ji Bisen Sir, Pro Vice Chancellor of Swami Ramananda Tirth Marathwada University, Nanded. Resource person of International E-Conference, Dr. Dinesh Dumar, postdoctoral researcher from France. Dr. Jagdish Mahale from University of Oxford, England. Dr. Prajakta Kulkarni, scientist and entrepreneur from California, America. Dr. Debashish Nayak, researcher from IIT Indore, and Dr. Rahul Kodgule, epidemiologist from Denmark Pharmaceuticals, Mumbai. On behalf of Dayanand Education Society, Dayanand College of Pharmacy, and Dayanand Institute of Pharmacy, I cordially welcome each and everyone present over here. At the very outset, I want to express my deep gratitude to Dr. Jogendra Singh Ji Bisen Sir for accepting our invitation to inaugurate the program in spite of his busy schedule and agreed to share the platform. Thank you so much, sir. Once again, I welcome you. I also extend warm welcome to our resource person for giving their precious time to share their expertise knowledge with our delegates. So, I am very glad to receive all of you at this virtual platform and to get the opportunity to hear you. I am grateful to Honorable Management of Dayanand Education Society, respected Honorable President Sri Lakshmi Ramanji Lahoti sir, Secretary, respected Honorable Rameshji Biani sir, Office Bearer Sri Lakshmi Ramanji, Sri Vishalji Lahoti sir, Office Bearer Sri Ajinkyaji Sonone sir, for permitting us to organize Grand Gala event, and they are actually the driving force for us. I also extend words of welcome to Dr. Sureshji Bhattad sir, who is renowned and senior pediatrician of Latur city. He is an instrumental for such activities conducted at college level. I always feel fortunate to be a part of Dayanand Education Society, which is one of the most reputed and pioneering education society in Marathwada region. Even since its establishment 1961, the society has grown from strength to strength by establishing different colleges and rendering human services to the educational needs of the youth. Our Dayanand Pharmacy colleges imparting quality education and maintain educational standards in all aspects like sports, research, cultural, and academic. Academic excellent results in university exam as a university ranker and national level competitive exams like GPAT, CMAT, and NIPER. This corona pandemic has wreaked havoc around the world, which has shaken the healthcare and pharma industry. These sectors are playing critical role in assisting India to overcome this difficult task. So it is our prime responsibility to focus, to respond, to receive and to provide efficient therapy in backbone of this situation. Due to this only, our college has organized international e-conference. During this conference, speakers will concentrate on COVID-19 diagnostic challenges and innovations, animal models, COVID-2 pathogenesis and transmission, nanomedicine approach, development of vaccine, and most important, efficacy and safety of COVID-19 vaccine. So, I wish this is very informative, fruitful, and productive and pleasant experience for all of us. So, keep learning, stay safe, stay healthy. Thank you very much. Over to Tiwari madam.
Thank you very much, madam, for your zealful welcome and for sharing your precious thoughts and for words of wisdom. Again, thank you. Now we are having Dr. Sureshji Bhattad, Management Representative, Dayanand Education Society. So, sir is guest of honor for our today's inaugural session. So, I request Dr. Sureshji Bhattad, sir. So, please give us opening remark.
Good morning, everybody. Today, I'm getting immense pleasure to be amongst you through this international conference arranged on COVID-19 era need and challenges in medical health workers and pharmaceutical workers. On behalf of Dayanand Education Society, I, Dr. Suresh Bhattad, warmly welcome all the eminent speakers, participants, pharmacy workers, students, and all those who are attending this today's e-conference. I convey my deep sense of gratitude towards our dynamic and vibrant President, Sri Lakshminarayanji Lahoti, and Secretary, Sri Ramesh Jibiani, who gave me chance for today's inaugural function. I'm getting great pleasure and it's a great privilege that Dr. Jogendra Singh Bisen, who is son of soil of Latur, is with us as a chief guest. He is son of soil of Latur, is associated with our institution since long duration, and he's having special love and affection towards our institution, and that's why we got a chance through this SRTR University of Nanded. I am also thankful for the great contribution to the Association of Pharmacy Teachers and to the Indian Pharmaceutical Association for their sponsorship. Today's selection of topic is a need of time. It's a need of time to our country and worldwide. And hence, I must congratulate and appreciate the efforts taken by our convener, Sakute Madam, Umbrella Sir, Coordinator Sonone Sir, Mr. Chaus, Lohia, and all other who are directly or indirectly associated with this. I'm pediatrician by profession. Hence, I will I will advise that you must vaccinate children on priority basis. From the list of eminent speakers, national-wise and international-wise, I noticed that all are of Indian origin and are settled at various places worldwide. They built up floors after floors in their academic career. They are highly qualified and they are giving their time to us. So please give big hand to them. Through this conference, we are bringing all the researchers, scientists, participants, pharmacological students to share their view and discuss their practical experiences. Now, this is the time to inaugurate conference. Hence, I declare that today's e-conference on COVID-19 era need and challenges to health workers and pharmaceutical workers, arranged by Dayanand College of Pharmacy and Dayanand Institute of Pharmacy, is inaugurated. I appeal to all participants to stay joined and gain the maximum knowledge and feast of knowledge through this conference. Last, I am thankful to Satpute Madam to drive this circular task. Without her team, it was not possible to arrange this large e-conference. I am also thankful to Mr. Chaus, Mr. Lohia, who gave me time and who make me arrange this conference successfully. Thank you.
Thank you so much, sir, for your influential message and your amazing speech and amazing inauguration of this conference. We are very grateful to have you here, sir. Now we are having Dr. Joginder Sinhaji Bisen, sir, Pro Vice Chancellor, Swami Ramanand Tirth Marathwada University, Nanded, as our chief guest for today's inaugural session. Sir is a true inspiration for this conference. Sir is always been source of motivation for us. We are very fortunate to have you here, sir. Now I request Dr. Joginder Sinhaji Bisen, sir. So please share few thoughts with us.
Good morning to one and all. Honorable Chair, President of Dayanand Education Society, Lakshminarayanji Lahoti, Honorable Secretary, Dayanand Education Society, Ramesh Jibiani, Dr. Sureshji Bhattar, Vishalji Lahoti, Ajinkyaji Sonone, Principal Kranti Satpute, Principal Srinivas Bumrela, well-renowned resource persons, other dignitaries, and dear participants. At the very outset, I take pleasure in congratulating the college and the team of organizers for initiating this international e-conference on the topic which is a greater relevance to the whole world. This pandemic has created a big challenges not only for our nation but also for whole world. Few countries of the world who were proud of their economical and ammunition power were to not able to handle this pandemic situation. Even these countries were inadequate to handle this virus. Since ancient times, many things such as turmeric and various spices were and still are the part and parcel of our day-to-day life. But we hardly realize the importance of these, even the significance of Holy Tulsi, which plays an important role in building of immunity, has been ignored. Our rich Indian culture consists of such several important values which have been ignored and neglected by us in the name of modernization. But this situation has made us realize the significance of these values. We must need to search and adopt these values. It is really unfortunate that even in the 21st century, we, the human race, when faced with such pandemic struggle to prevent our population. But if one looks carefully in this issue, we know that this virus does not differentiate between the rich and poor, the rural and urban also. Hence, we must leave all our egos and follow humanity and behave accordingly. Now it is our duty to face this problem with confidence and consider this problem as an opportunity to build our nation and our
Thank you, sir, for sharing great knowledge with us. Thank you for your words of wisdom. So there is some technical issue with Pro VC sir's network. Sir will shortly join us. Please wait. Sir is joining shortly. There is some technical issue. So stay tuned and stay connected with the international e-conference. So we are having 2 days international e-conference on COVID-19 era, need and challenges in healthcare and pharma sector. I request now Pro VC sir to please again rejoin the meeting.
Sorry for inconvenience.
It's all right, sir. No issues, sir. You can continue, sir.
As we all have seen several critical conditions to fight against contagious disease. Now it is high time to cope up with this serious condition. I am sure in this e-conference, much deliberation will be take place in the resource persons talk. This conference will provide important inputs to the nation and world. I once again congratulate the organizers and wish best for deliberation. Lastly, I would like to just say, मन में है विश्वास, पूरा है विश्वास, हम होंगे कामयाब. conclude with my song of Chirag Jain. थम गया संसार सारा, हो रहा ओझल किनारा, सांस डर कर ले रहे हैं, विष घुला है हर नजारा, किंतु हम खुद को पुनः सुकरात कर देंगे, फिर नई शुरुआत कर देंगे। मानते हैं आदमी से भूल भारी हो रही थी, मानते हैं आदमी से भूल भारी हो रही थी, एक अंधी दौड़ की सब पर खुमारी हो छा रही थी। इस हवस का अंत होगा, इस हवस का अंत होगा, होड़ का अवसान होगा, फिर नई दुनिया बनेगी, नए सृजन का गान होगा, खेत बनकर सबका सही अनुपात कर देंगे, फिर नई शुरुआत कर देंगे। लोभ का प्रतिकार करना जानता है वो बचेगा, लोभ का प्रतिकार करना जानता है वो बचेगा, सत्य को स्वीकार करना जानता है वो बचेगा, जो बचेगा वो मनुष्यता के लिए वरदान होगा, जो बचेगा वो नए युग का नवल उत्थान होगा। साथ मिलकर फिर नए हालात कर देंगे, फिर नई शुरुआत कर देंगे। थैंक यू।
Thank you so much, sir, for sharing great knowledge with us and for your words of wisdom. Your thought-provoking address and lines already has made our day wonderful. So thank you very much again. Now we would like to conclude this inaugural session. My request, Ms. Rajmane, madam, please introduce our eminent speaker.
Thank you, Shraddha ma'am. We are starting with first session. It's the matter of immense pleasure to introduce one of the versatile personality, Dr. Dinesh Dhumar sir, postdoctoral researcher at CNAM, Marseille, France. Dr. Dhumar sir has completed Bachelor of Pharmacy and Master of Pharmacy from North Maharashtra University, Jalgaon, India. Sir was formerly working as assistant professor at R.C. Patel Institute of Pharmaceutical Education and Research, Shirpur, India. Sir was honored with his PhD in the year 2018 from Institute of Chemical Technology, Mumbai, Maharashtra, India. Honorable Dhumar sir has received prestigious Raman Charpak Fellowship in the year 2017 at CNAM, ex-Marseille University, France. At present, Honorable Dhumar sir is working in the area of nanomedicines under the supervision of Dr. Ling Peng. Sir has glorious pharma career. He has various distinctions to his credit. Some of them are Best Oral Presentation in
Sorry for the interruption. I extend my vote of thanks to all the dignitaries for coming to the inaugural session. I thank President Dr. Lakshmi Ramanji Lahoti sir, Dr. Rameshji Biani sir, Dr. Sureshji Bhattar sir, Dr. Ajinkyaji Sonone sir, all management members, Dr. Joginder Sinhaji Bisen sir, Pro Vice Chancellor Swami Ramanand Tirth Marathwada University, for coming here for the inaugural session from their busy schedule. So from Dayanand College of Pharmacy, on behalf of Dayanand Education Society and Dayanand College of Pharmacy, I thank all the dignitaries for inaugurating this international e-conference. Thank you very much. Here we conclude with the inaugural session. Thank you. Before starting with the technical session, we are announcing to all the participants that if they are having any doubt, any issue or any question, then they can please raise your hands on the YouTube, or you can type your question in the chat box, and the eminent speaker will solve your queries and your questions after the completion of technical session. Now I request Trupti Rajmane madam to please introduce our eminent speaker.
Thank you, Shraddha ma'am. We are starting with first session of this international e-conference. It's the matter of immense pleasure for me to introduce one of the versatile personality, Dr. Dinesh Dhumar sir, postdoctoral researcher at CNAM, Marseille, France. Dr. Dhumar sir has completed Bachelor of Pharmacy and Master of Pharmacy from North Maharashtra University, Jalgaon, India. Sir was formerly working as assistant professor at R.C. Patel Institute of Pharmacy, Shirpur, India. Sir was honored with his PhD in the year 2018 from Institute of Chemical Technology, Mumbai, Maharashtra, India. Honorable Dhumar sir has received prestigious Raman Charpak Fellowship in the year 2017 at CNAM Aix-Marseille University, France. At present, Dhumar sir is working in the area of nanomedicines under the supervision of Dr. Ling Peng. Sir has glorious pharma career. He has various distinctions to his credit. Some of them are Best Oral Presentation, International Travel Grant, winner at various international conferences, gold medalist in Master of Pharmacy, academic scholarship from Sir Ratan Tata Trust, etc., etc. Sir has deep research interest in the area of nanomedicine for drug delivery, gene therapy, immunotherapy for cancer and infectious disease treatment. Dr. Dhumar has published 20 research papers. He has published one book and filed one patent also. Today, he is going to address us on the topic nanomedicine approach to combat COVID-19 pandemic. I would like to request Dhumar sir to take over the charge and address us. Honorable Dhumar sir.
Thank you. Thank you for your kind introduction. And I would like to share my presentation and I will start. Is it okay?
Yes, sir.
Okay. Thank you. Can you see my slides? Hello? The slides are
Yes,
visible?
yes, yes, sir.
Okay.
It is visible. Yes, sir.
Okay. Thank you. Thank you, ma'am. So my topic, the title of my topic is the nanomedicine approach to combat COVID-19 pandemic. As in the inaugural session, Pro Vice Chancellor sir also mentioned that how COVID pandemic has devastated our world and our country also, and how we have suffered through this disease or the pandemic from last 1.5 years. And I'm going to talk about how nanomedicine helped in combating this COVID-19 pandemic. And basically, nanomedicine has provided us help in the initial stages of the COVID-19 pandemic, having the mRNA COVID vaccine, which are based on the nanomedicine. And my talk will be based on the COVID vaccines based on mRNA technology. So the content of my presentation will be like, I am the one who is starting this conference. So I would like to throw some light over the COVID-19, how it is the structure of virus, the life cycle, how the transmission of coronavirus occurs. Then what are the therapeutic possible strategies for COVID-19, how the management so far we have done for it. Next is the nanomedicine based strategies. As I know, the audience must be some students also, faculties also. So I would also like to throw some light over what is nanomedicine, what are the classes application, and how this nanomedicine helped us in a COVID-19 era. And next, the mRNA and development of mRNA vaccine, which is based on the nanomedicine technology. So first, structure and life cycle of the COVID-19. As you see in the figure, like the COVID structure is similar to the most of the viruses with extra spike proteins on the surface. Then it has a membrane glycoproteins, there is envelope protein, and it also contains a single-stranded RNA, which replicates for building the new copies of the virus. So and this virus can be spread through water, air, food, blood, touches, and insects. So the life cycle of SARS-CoV, how it goes, like the COVID virus comes into the cell, it inject the viral RNA into the cell. In the ribosomes, the machinery, the protein gets replicated, and the replication of viral RNA and synthesis of viral protein goes there. So the assembly of new virus forms, and it releases, and death of the infected host cell. So this is how the cycle goes. And you can see on the screen, there is a transmission electron microscopic figure of the SARS-CoV virus, and we can see clearly how the spike proteins are on the surface of the virus. So these spike proteins has played a really important role for this COVID infection, because with the help of this spike protein, the virus attached to the host, and then it enter into the cell. Now I will explain how it attaches and how the cycle goes on. So if you see in the figure, like how the viral factor and the host factor responsible for this pandemic actually. So we all know it is originated from bats. So, and some theories believe that it went from bats to human directly, and some theories believe that it went from bats to animals, domestic animals, and then into human. So from bats, when it attaches to the human, how it goes. So for the human, it attaching to the host receptor ACE2 enzymes, including two subunits, S1 and S2. So S1 determines where the host range and cellular tropism by binding it. And second, S2 mediate virus cell and membrane fusion by HR1 and HR2 protein. Similarly, in terms of the domestic animal, it has a similar binding like human cells, like it binds to the ACE2 an enzyme in swine, civet, but not in case of mouse. So the M protein is responsible for the transmembrane transport of nutrients that bird release and formation of the envelope. So every protein shell plays important role for the COVID virus replication. So N and E protein are the several accessory protein interfere with the host immune response for unknown function. Other known molecules facilitate membrane investigation and the cofactor. So these are the factors for the virus, how every protein component of the virus is responsible in its replication. So in terms of the host factor, so SARS-CoV-2 receptor, like it's a human angiotensin converting enzyme 2, it attaches. Next, individuals who are susceptible to severe diseases, like elder people who are more than 65-year-old, people with the underlying diseases, such as you can see on the screen, hypertension, chronic obstructive pulmonary disease, diabetes, and the cardiovascular, who are already have these kind of diseases, are more prone to these infections side effects. So when this infection happens, there is a cytopathic effect occur and the cytokine storm. And this cytokine storm is mostly affecting the patient, which has an underlying cause, which I mentioned. So these inflammatory responses, then hypoxia, there is a septic shock, etc., may related to the critical condition. And because of this condition, the patient suffer a lot. And we have seen that in a treatment, we don't have treatment for this in infection. So we use symptomatic treatment based on the different symptoms. So this leads to the severe complications, like as I mentioned, like because of the cytokine storm, we have respiratory distress syndrome, there is a septic shock, metabolic acidosis, hard to correct. There's a coagulation dysfunction leads to the heart failure. Then there is multiple or organ failure. So these are the host factors. So viral factor and the host factor both are responsible for this devastating pandemic. And if we want to treat, we need to study these all factors. For killing or stopping the virus, we need to learn the viral factors. And to treat a patient, if we don't have medicines currently, the host factors, we need to keep them in control. And we should give some symptomatic treatment. So what are the therapeutic strategy possible for a COVID-19? So if you see the cycle of the virus cycle, based on the different steps, we can achieve different therapeutic approaches. Like first, the COVID attaches to the host cell through the enzyme ACE2. So we can think of virus attachment inhibitors by blocking the ACE2 receptors. Then the virus goes into the cell, into the cytoplasm, then it goes to the endosome, then there is a lysosome, and it follows the cellular machinery to release its internal protein matter, the single strand RNA, which replicates and which goes into the ribosome machinery to create new copies of it. So at every step, we can have some therapeutic approach. We can develop virus inhibitors. The research towards this is going on. Then we can have a lysotropic agents. Then we have a RNA replication inhibitor like remdesivir was used highly in India, which is still not clinically approved in Europe or the other countries, which is RNA polymerase inhibitor. So which helps to stop the replication at that point. So these kind of inhibitors are still in a clinical trial to use. Then we can use a protease inhibitor. Then there is a neuroanimidase inhibitor. So likewise, we can use different strategies to combat. Next, when this virus replicates, it has a pro-inflammatory and cytokine reactions in the body, which releases various inflammatory mediator, macrophages, and which lead to this cytokine storm. And this cytokine storm was mainly responsible for death of many people whose body reacted really devastatingly to this. And because of the unavailability of treatment to kill the virus, which lead to the death. So we can target specific inflammatory molecules, immunomodulatory therapies and cellular immunotherapies. So these are all probable approaches we can think, and which are under studies. Some of them are in clinical trial, but they are not actually in a treatment for by the FDA approval or the WHO approval. So till now, how we have done the management for the COVID, like we understand. So we suggested, we tried protection using masks and personal protective equipment, which actually helped to stopping the virus transmission. Then imaging helped, especially in India, because in some cases PCR was not able to detect the infections in many patients. So some imaging techniques help us to understand how the virus goes deep down into the lungs. Next, detection. I will not go in detail for the detection, as I know some other speaker going to talk about it, how nanomedicine also help in detection of the COVID, and the detection leads us to help in a further management of the disease. And next is the treatment and vaccination. So various drugs are in clinical trial, vaccines are currently in a market. Then the newly developed drugs will for sure need delivery systems to target in lungs, and there are oxygen therapies are on the way also. So I will mostly focus on talking about the vaccine, which are nanomedicine based. But before going to the vaccine, which are based on the nanomedicine, I would like to throw some light on the nanomedicine. So what is nanomedicine? Nanomedicine is a branch of medicine that applies knowledge and tools of nanotechnology to the prevention and treatment of disease. So nanomedicine involve use of nanoscale materials, such as biocompatible nanoparticles. There are nanorobots, and these can be used in various applications for the diagnosis, delivery, sensing, acquisition purpose in a living organism. It has a various application. So till now, nanomedicine has helped in various causes, especially in a cancer treatment also. So knowledge of this background are really essential when we want to do some research. So with the help of nanomedicine, like we have achieved overcoming barriers. Like we have done the intracellular deliveries, navigating to micro or environment, targeting immune cell, crossing epithelial barriers. Vast of research from last 2 or 3 decades was there on nanomedicine, and obviously these experiences help us when we have to challenge to new diseases. So we need to dig into the backgrounds, what we have done till now, what we have achieved, what we have what we have understood based on the different delivery systems before we have used. So there are different classes of nanoparticles we have studied before. Like there are polymeric nanoparticles, there are polymorphism, there are dendrimers. In my group here in France, we work on mostly dendrimers. Then polymeric micelles, nanospheres. There are inorganic materials also, like silica nanoparticles, quantum dots. There are iron oxide, gold nanoparticle had played a huge role in detection, diagnosis, and treatment also. And another important class is lipid-based drug delivery system, which I will talk because the new mRNA vaccine are based on the lipid drug delivery system. So in that we have liposomes, lipid nanoparticles, there are emulsions, there are micelles, there are many. Here I'm mostly talking about there are various multiple subclasses, but here I'm talking mostly about some major classes, and each class has like a numerous broad advantages and disadvantages regarding the cargo delivery and patient purpose. So recently a new term have came in terms of nanomedicine is called as a personalized nanomedicine, where we talk about the patient history, patient disease progress. Based on that, we work on the structural aspect, and based on that, we develop the nanosystem. That is a different class to talk. So these all nanoparticles has a added advantages and disadvantage over each other. Like polymeric nanoparticle, they're precise to control of particle characteristic. They have payload flexibility for hydrophilic and hydrophobic cargoes, easy surface modification, possibility of aggregation, and toxicity is the disadvantages. Then for in organic materials, they're unique electrical and magnetic. They have unique electrical, magnetic, and optical properties. Their variability in size, structure, and geometry helps well suited for diagnostic applications, and toxicity and solubility and are the limitations for it. So the formulation simplicity with the range of physiochemical properties, high bioavailability, payload flexibility, and low encapsulation efficiency is a disadvantage for lipid-based drug regulation. So here we see we have advantages and disadvantages, and we have to work on them for selecting suitable class for a particular application. So commonly engineered nanoparticles for surface properties that allow for enhanced delivery. So till now we have seen that in a nanomedicine we can engineer them. We can modify their surface according to the need. We can add the targeting agent like which antigen or antibodies or cell surface receptors. We can add aptamers and peptide for a particular receptor. We can change the architecture like site, shape, and charge, because for every application, these architectural changes help a lot. Then we can do pegylation for a stealth approach where the long circulation in the body can be achieved. There is cell coating, density, and toughness. So these are various things we can alter, engineered on a nanoparticle, which help in to achieve our goals and needs. Until now, various FDA approved nanomedicines are in market. And nanotechnology has definitely helped humankind and pharmaceutical industries to achieve their targets and goals in a well manner. So, like just to give an example, currently in India, we had a mucormycosis, had a problem, and the ambisomes or the fungus
पुढचा टॉपिक आहे
is the
ना,
fungal
आता आम्ही
or
शिकूया
the
ना ते टॉपिक. अँटीव्हायरल.
I can somebody stop their microphone? Hello. So like as I'm explained, like lipid-based drug delivery system helped a lot. Like as I explaining, like ambisome currently in India, mucormycosis, which is a black fungus, and the treatment for it is again a nanomedicine, which is ambisomes, which are the liposomes of the amphotericin B. And recently, the first siRNA drug delivery system which came into the market, which is approved by FDA for the amyloidosis in 2018, and which has actually led to way for mRNA vaccines also. Sorry. And there are polymer-based drug delivery systems are there. And there are so there are various FDA approved nanomedicine. So what this story tells us that nanomedicine actually help us in combating certain diseases in a precision manner, and which helps us better in terms of the conventional therapies. Sorry. Okay, so next is how the nanomedicine help in the infection, mostly the respiratory infection. Like, and there are various viruses which responsible for the respiratory tract infection. Coronavirus is one of the example. Then there's the herpes virus. Then there is mammalian influenza virus, rhinovirus. Then there is adenovirus, respiratory symptoms like influenza virus. So there are various viruses which responsible for the respiratory tract infection. And again, these all infection leads to the cytokine storm, which lead to the alveolar death and some inflammatory responses in the body, and which leads to the severe patient conditions and death also. So various nanoparticles, they have features which can help us to overcome these diseases, because they have ability to increase solubility of hydrophobic drugs. Like hydrophobic drugs is difficult to reach to their target site or efficacy due to their hydrophobicity. So the nanomedicine helps us to reduce their increase their solubility. They have high adhesive capacity, quick mucosal penetration, which is actually necessary for the COVID infection also. Then there is targeted delivery to bronchi, alveoli. Then there is direct interaction with virus, the modulation, then prolonged mechanism action. So these all leads to the, these all helps for the treatment of this kind of diseases. So what are the different strategies we can use for COVID vaccine? Sorry, for the COVID treatment. So there are two approaches we can think now based on the nanomedicine. The first is nanocarriers encapsulating antigen or gene, or the nanocarriers having antigen on the surface. So first is like delivery towards the antigen representing the cell which cause immune reaction, because the best import, like we can have a two treatments. First, the antiviral drugs, and second, host immune system response. So vaccine comes under the home immune response. So in that, what we can do, we can have a nanocarrier encapsulating antigens, DNAs or RNAs, and that we can deliver, and which can activate the antigen presenting cell, and which lead to the activation of the human immune system. Second, in that we can have a two strategies I mentioned: encapsulation of the cargoes inside, or second, we can have antigen on the surface of the nanomaterial. Second strategy which are under development, and some of them are in clinical trial, which is not in a public domain. They are use of the targeting to the ACE2 inhibitor. So they target the receptor, either they block it using peptide or monoclonal and antibodies, and they have inside the drugs, as well as they only stop the enzyme. So the virus attachment to the enzyme stops, and it cannot enter into the cell. And this is mostly looking for the targeting nasal mucosa. But this strategy can be used for the secondary target sites like epithelial cell, intestine cell, or the central nervous system, where possibly the infection can spread. So these strategies can be used, and the vaccines are based, the mRNA vaccines are based on the first strategy for delivering or encapsulating the gene inside and activating the human system by delivering it to the targeted cells. So before going to the exact explanation how these vaccine have been formulated, so the two best mRNA vaccine which are FDA approved are the BNT162b2 from BioNTech and Pfizer. So BioNTech is a German company, Pfizer US based company. And second is a Moderna, which is again used mRNA 1273, which is from USA. So the BioNTech, Pfizer, and the Moderna has like similar mode of action. So how? The vaccine has a nanocarrier with encapsulated mRNA. It goes into the human cell. Then the cell expresses spike protein. The spike protein, as I mentioned, it is responsible for sensing the human cell, and they attach and they expresses the immune response. So if our cells expresses the immune response, is built up there. So mRNA encoding for the spike protein is protected in a lipid nanoparticles here. And I will explain why they require protection. And once absorbed, the cell expresses and resulting in an immune response. So the efficacy of mRNA vaccine for the Pfizer is 94 to 96%. It need like 2 doses, 21 days apart. And the problem with this vaccine is a cold storage, like minus 70 degrees Celsius for 6 months and 2 to 8 degrees Celsius for 5 to 7 days. And similar mechanism action is for the mRNA vaccine also from the Moderna. It acts same, and it has a 94% efficacy. And it has a 2 dose, 20 days apart, and the storage for this one is minus 20 degrees Celsius for 6 months. So delivery gene using lipid nanoparticle, it has a low immune response, no genome insertion, increased gene size, lower potential anti-vector immunity, and cell-free manufacturing. Whereas in terms of a conventional vaccine, there is always the required cell culture. Like the two vaccines available in India, they are traditional vaccine, like the Bharat Biotech vaccine, where they use the inactivated virus, where they are chemically inactivated with a chemical called as a beta prolactin. So it cannot replicate, but all the protein remain intact, and this protein generate the immune response. And similar vaccine where in terms of Oxford or AstraZeneca, we call it as a Covishield in India, as a viral vector, the adenovirus vector, where double-stranded DNA encoded for the spike protein is protected in a safe viral cage, and the spike protein which leads to an immune response. So the AstraZeneca vaccine or the Covishield has like 82% efficacy, and for Covaxin it is 81%. And they also need two doses. So now I will go for mRNA, like what is mRNA? So messenger ribonucleotide, mRNA, is a single-stranded molecule of RNA that correspond to genetic sequence of gene and is read by ribosomes in the process of synthesizing protein. I will not go in a detail like how they sequence engineer mRNA or how they modify mRNA according to the need. But I would like to explain that the mRNA are so specific for a specific DNA to generate a specific protein. So here these mRNAs are engineered to generate the spike protein in a host, where then our body can have immune response to it. And the structural element mostly is that the in vitro transcript IVT mRNA technique used these days, which is really efficient for in engineering the mRNAs. And as I mentioned, sRNA are the first component which came in FDA approval, and then the mRNA. So the mRNA's molecular weight is like 10 as to 6 ratio, and whereas sRNA are small as compared to that. mRNA are mostly a single stranded. They have 5' and 3' end. The 5' cap and the poly A tail as a 3' end. So why mRNA need protection? Because unprotected mRNA are vulnerable to the enzymes like nucleases. So they get degraded if they are not protected. If you inject just mRNA, our blood has an enzyme and that enzyme degrade. Another, they have a polyanionic nature. And our cells also has a polyanionic nature. So there is a repulsion. So they cannot internalize easily into the cell. So they need a delivery system so they can reach into the cells in their efficacious amount. So where the lipid nanoparticle plays important role. So how lipid nanoparticles protect it. So I will first explain the construction of the lipid nanoparticle and what are the components used. So here we can see that the lipid nanoparticle has a different component. First, ionizable cationic lipid. So these binds and protect rRNA. They facilitate endosomal escape, because if our material doesn't escape from the endosome, it will go to the lysozyme and it will go out of the cell and it will not reach to the cell machinery. So it is important for it to have an endosomal escape. And they have a neutral charge at a pH 7.4, which is really important. Then they use cholesterol, which is important for endocytosis via LDL receptor and ApoE receptor recognition. Also, they give stability to the lipid nanoparticles. Then they use a helper lipid like DSPC, which helps for maintain the structure, and they use stabilizer to stabilize particles and protect them. And the major component is a PEG lipid, which gives a stealth approach, which helps it into circulating longer time in the blood. And these nanoparticles prepared using the microfluidic technique, where the mRNA are dissolved in aqueous phase and lipid are dissolved in ethanol alcohol phase. And they use the microfluidic technique where they go through different cycle, and at the end we have the lipid nanoparticles, and which have we have to use different purification techniques at the end, like dialysis and other, to remove the ethanol from it. And these are the structures of the lipid they have used in the construction of these liposomes, the lipid nanoparticles. So the ionizable cationic lipid has a distinct pK, which helps it to interact with the negatively charged mRNA, as well as it gets protonated when it comes into the endosome, where it get proton sponge effect, where it bursts the endosomes and comes into the cytosol. So, but when you develop these delivery systems, you need to have a different characterization technique to characterize them. So in terms of characterizing the lipid complexes, we use dynamic light scattering to check the particle size, which is really important to check the batch to batch variability and stability. Then the zeta potential or check. Then we check particle size with the NTA, TRPS. Then we have size exclusion, HPLC, for particle size distribution, assessing bound, unbound mRNAs. Then we use a different microscopic technique like TM, EACM, AFM to check the morphology, as I shown in a figure how they look like here. They have the circular bell shape, which helps. This microscopic technique helps us to know their size and morphology. Then we do many chromatographic assays like reverse HPLC, size exclusion, HPLC, then ion pairing, and etc., and the mass spectroscopy for the quantification of integrity of lipids in the mRNA. So these all parameters are really essential to check the stability and integrity of these lipid nanoparticles. So Similar, we have assay to monitor the mRNA drug substance inside. Like we should know whether the mRNA is stable and active inside. So sets of characterization, DNA templates and RNA transcript runs to check the identification, the purity, the molar mass, quality assessment was done. Then there is a characterizing mRNA and encoded translation going on. If we see previously in vaccine innovation, most vaccine takes year to develop, but scientists created multiple vaccine for SARS-CoV within 1 year. So how the technology or the science has gone up. Like if we see from typhoid fever to the SARS-CoV, every time sometimes it takes decades for us to get to the vaccine. But we are lucky enough that in this scientific era, we had the COVID last year, and within 1 year, we have achieved the vaccine. And in terms of mRNA vaccine, I would say the research in mRNA domain or the nanomedicine last 1.5 decade has helped to get this mRNA vaccine within 1 year. And the drug firm like Pfizer, BioNTech got their joint SARS-CoV vaccine approved less than in 8 months after the trial started. This rapid turnover was achieved by overlapping trials, and because of they did not encounter safety concern. So the nanomedicine helped the research, the background, the mRNA research helped us to get the vaccine within 1 year. So definitely nanomedicine helped here us. And that's why if you see, this is the only 2 nanomedicine based products approved within one year. So it tells us the importance of nanomedicine here. So these are the list of COVID vaccine using the lipid nanoparticles, which are in clinics and the clinical trials. As I mentioned, like the first by Moderna and second by Pfizer, they are in market, and there are various other vaccines using lipid nanoparticles are in a clinical stage or in the evaluation stage. In different countries with different academic institutes as well as different healthcare firms. So thank you. If you have any questions and comment, we'll welcome. And if you have more question, if I'm not able to answer here, you can contact me on my email or WhatsApp. Hello.
Thank you, sir.
Hello.
Thank you, sir, for your valuable insights on this topic. Sir has put a vivid picture of life cycle of COVID-19 virus, various therapeutic strategies for COVID-19 treatment in front of us. We came to know about different new approaches like personalized nanomedicine, nanocarriers, targeting of ACE2 inhibitors, etc. through this session. On behalf of our participants, I wish to ask a few questions.
Yes, ma'am. Please go ahead.
Is higher antibiotics which are used for COVID-19 treatment are responsible for mucormycosis?
No, for mucormycosis, mostly responsible are the steroids, which are used to reduce the inflammation. Because when you use excess amount of steroids, they reduce immunity in a patient. And because the role of steroid is to block the immune response or the inflammatory mediators, which leads to the inflammation of the lungs or the alveoli. So excess use of steroid stops this in inflammatory mediators or the immune system. So they stops. Because of this, when another species like fungus attacks, our system, the self defense system is not sound because of the use of excess of steroid, and which leads to the fungal infection in severity. Those people who didn't use steroids or has a good sound immune system, they will never had a problem, or they didn't had a problem for mucormycosis. But the immunocompromised patients with excess of steroid had a problem, and antibiotic will not lead to these kind of thing, but for sure excess of use of antibiotic definitely will create resistance, and they might create another superbug also. So I always advise take an antibiotics with the physician's prescription, just not by yourself or the over the counter.
Thank you, sir. One more question is there. How many zoonotic coronaviruses are reported till date?
There are, like, if you see recently, I would say for this zoonotic, the first coronavirus came in like in 2002 to 2005 when the first SARS come, then the MERS come, and I would say like there are more than 8 to 10 they have reported till now.
Okay. Sir, can you tell why 2 doses of the vaccine are needed, and on what basis the duration between 2 doses of vaccine is decided?
Okay, like this is mostly goes with the clinical data and how clinical trials goes on. And for this coronavirus, we doesn't want to go give a high dose in the first one, because sometimes if you go really high dose of the vaccine, the immune response will trigger, and some patient might have anaphylactic shocks where it will be difficult to treat. So 2 doses given on. The first dose is a light dose and the second dose is a heavy dose. With the first dose, we still get like a side effects, like there is a swelling in the arm and something. And with the second dose, we get a fever, there are rashes, chills. Some people had severe side effects of the coagulation in terms of the AstraZeneca vaccine, and some had a paralytical attacks and all. So based on the patient history and thing, the dose concentration is decided. That's why here they decided to go for two doses, one light and one high dose. And for the Pfizer vaccine, initially they decided 21 dose, later 21 day dose, later they went to the window of 39 42. Is based on how immune response react to the two different doses, and the duration window is decided based on the clinical trial history and the current patient history for the better immune response.
Okay, thank you, sir. One more question is there.
Yes, please.
Can we use nanotechnology in the treatment of black fungus?
Yes, that's what I said. We are using nanotechnology actually for the black fungus. The amphotericin B injections we are using, these are the liposomes already. And just to tell, like even in our lab, we started working with the amphotericin B now to prepare dendrimer-based particles also to target this kind of a fungus. So yes, nanotechnology definitely used to treat this fungus. The reason is that this fungus spread to the brain, eyes, and the drugs are not what, like, the only treatment for this is amphotericin B. And amphotericin B is not water soluble, and you cannot take it as it is. So we need to make it water soluble, and the nanotechnology help us to solubilize this hydrophobic drug. And with this, we can also target the fungus using targeting agent. Like if the fungus reaches to the brain, it is difficult for a normal nanotechnology drug delivery system to cross the brain because of the blood-brain barrier. So various approaches are going to target the blood-brain barrier by using peptides or the nanobodies or a targeting agent to cross BBB. So if the infection spreads to the brain, it's more devastating because it leads to the death of the patient. So for definitely nanotechnology helps in treating these kind of diseases.
Okay. Sir, can you highlight on Covishield vaccine, which is causing blood clots?
It's like I have read even yesterday. I was reading with the Pfizer vaccine also, but for the COVID vaccine, it's not in all the patients. In few patient group, they are having a response. As I know, like even in a COVID patient, not the vaccine, some patient had blood clotting and heart attack, which is cause of the death. So I think similar reaction is triggered with the people who are taking Covishield, where they have a blood clot or blood coagulation effect. This is not for all patients, but certain group of patients where their immune response trigger this coagulation, and this clots leads to severe side effects.
One last question is there.
Please.
Are monoclonal antibodies give immunity sooner than vaccine? Which is to be preferred? Monoclonal antibodies or vaccine?
Like monoclonal antibodies or vaccine, I would say that I say like this is a topic of debate, and this is depend also. Like if we say monoclonal antibody, then again we need to target them. That is really important. And in terms of normal vaccine, I would say mRNA vaccines are more advanced as compared to the monoclonal antibodies, and they create more response as compared to the monoclonal antibodies.
Thank you, sir. It was a wonderful, informative session. On behalf of our whole team, I would like to thank you, sir.
Thank you very much for giving me opportunity to talk on this in Copan conference. I would also like to thank Gopal for in inviting me to give talk in this e-conference. Thank you very much.
Thank you, sir. Thank you very much.
I will stop sharing.
Yes.
Thank you.
Thank you, sir. After this informative, enthusiastic, and energetic session by Dr. Dinesh Dhumar, sir, all the participants are eager to listen to the next eminent speaker, Dr. Jagdish Ji Mahale, sir. I would like to invite Mr. Gopal Lohia, sir, Assistant Professor, Dayanand College of Pharmacy, to introduce next eminent speaker, Dr. Jagdish Ji Mahale, sir. Over to Lohia, sir.
Thank you, madam. Good afternoon to one and all. Myself, Gopal Lohia, Assistant Professor, Dayanand College of Pharmacy, Latur. Welcome you all for this second technical session of this International E-Conference. For this session, we are having speaker Dr. Jagdish Mahale, postdoctoral research associate, University of Oxford, England. It gives me an immense pleasure to introduce our speaker. He has completed B Pharmacy from Savitribai Phule, Pune University in the year 2009. He has completed Masters in Pharmacology from North Maharashtra University, Jalgaon, Maharashtra in the year 2011. He has completed PhD in the Cancer Studies and Molecular Medicine from the University of Leicester, UK in the year 2016. He worked as a clinical trial coordinator at S7 Clinical Research Center, Nashik, Maharashtra. He is having the experience as a drug safety associate at Roche Pharmaceuticals, Mumbai. He also worked as a pharmacovigilance scientist in the TCS. His expertise in handling various animal and immunological models, also having research skills in vitro, in vivo, and ex vivo studies. He has published various international research papers in journals of high repute and good impact factor. Presently, he is working as a immuno-oncology researcher at University of Oxford. With this short introduction, I welcome the today eminent personality, Dr. Jagdish Mahale, to talk on the topic development of Oxford AstraZeneca vaccine and further studies. So I request Dr. Jagdish Mahale, sir, to give the talk. Welcome, sir.
Thank you very much, Gopal, for your kind introduction. Am I audible to everyone?
Yes, you are audible.
Okay, I will just start sharing my screen. And is my presentation visible to everyone?
Yes, the presentation is
Okay.
visible, no problem.
Excellent. Okay, thank you very much, Gopal, for your kind introduction, and thank you for inviting me to give this talk. So, as Gopal said, I've been working at Oxford since 2016 after completing my PhD in Leicester. And when I was given the invite to present a talk at this e-conference, I thought it was befitting because I come from Oxford and it was a COVID talk that I talk on the vaccine that was developed by my university. So I'll just give, so my talk is like less scientific and more like a story. So I hope everyone enjoys it. Just a second. Okay, so theme of my talk. So I'll just start by going into a background and history of not just the vaccine, but the institute where it's developed, because it has got a very historical importance in terms of how where it was developed and how the institute came into place in the first instance. Then I will talk about the paper discussion, like the Oxford paper, the Lancet paper that was published in December 2020. Last year. It won't be a boring paper discussion, I will assure you. It will just be a very quick snippet of important findings and some of the controversy that had arrived after the paper was published and even after when the vaccine was approved for general public use. I will share some light on that. This will be a very short part of my talk, further studies and treatment, but I will try and explain as much as possible, talk about what's the post-COVID era actually, like, which is one of the theme, and how will we ever come out of this pandemic, and how would the pandemic be, how would the post-COVID life be, and will we ever face another pandemic, and when, how, when, or will we ever have another pandemic? So I'll just start by talking about the institute where the Oxford AstraZeneca vaccine was developed. So this is the building that you see in the green. That's the Jenner Institute where the Oxford AstraZeneca vaccine was developed. And the building over here, you see, the next one is the Kennedy Institute where I work. So it was quite a moment of pride and joy. Is the vaccine, which is saving millions of lives all over the world, was developed in a building right next to me. So I just wanted to show you where exactly the Jenner Institute is and why it's called the Jenner Institute. If you all know, the institute is named after Edward Jenner. Edward Jenner is often considered as a father of immunology, and he was the first person to develop world's first ever vaccines against smallpox. And if you go into the story of how he developed smallpox, it was just by a mere simple observation. Smallpox was one of the very deadly diseases in England around that time, or not just England, everywhere. And I think around 20% of the people who got infected with smallpox, they used to die. Edward Jenner made a very simple observation. The milkmaids who used to always be in contact with the cows, they never got smallpox, whereas other people used to get it. And he was thinking why it must be. His simple observation was cows have got cowpox. And because the milkmaids are in frequent contact with the cows, they develop some sort of immunity because the cows are always infected with cowpox. So he did a very simple experiment. He took a smallpox live virus and he just infected a 14-year-old or 15-year-old boy. He developed a reaction. He was monitored. Is okay. He covered, and then he tried to infect again the same boy with smallpox virus, and he didn't develop smallpox virus. A very simple experiment, a very simple observation, and that led to discovery of world's first ever vaccine against smallpox. And this scientist, this English physicist, English physician, has been credited by single-handedly saving most lives ever. And the good thing is, he was just he did all these experiments in and around Oxfordshire. He also worked for a small time in University of Oxford. So that's why the institute is named after eminent great scientist Edward Jenner. So this Jenner Institute, which actually developed this vaccine, it's a very well-known, respected institute in the field of immunology, in the field of vaccinology. It has already successfully developed vaccines against already various other forms of viruses, coronaviruses like MERS, SARS, tuberculosis. Their recent successes, which was a vaccine against malaria. This very soon be again will be in the market. It got a phase 3 results were out few days ago, and it's provide up to 75% efficacy. So this Jenner Institute in itself, it's a very successful institute in developing vaccines against various forms of viruses. So they developed a vaccines against corona in such a short span of time. Isn't surprising because they had all the technology and they had all the resources needed. All right. So how did they came into developing this vaccine so fast? My previous speaker, my friend Dinesh, he already spoke about how what's the design and what's the principle behind the vaccine. So I'll just quickly go through this again. So this is the coronavirus. Okay, so why is it called coronavirus? So it is studied with the spike proteins on the surface, right? And these spike proteins, when you observe this virus under a microscope, it looks like a crown. And corona is a Latin word for crown, so that's why it's named coronavirus. And so it expresses the spike protein on its surface. What do the spike proteins do? The spike proteins is actually a way for this virus to enter the host cell. And obviously the spike protein is expressed by a spike protease gene. So to deliver a vaccine, what Dinesh was obviously previously speaking, you need some form of delivery that will carry this spike protein into a human body. And what Oxford Zeneca vaccine is developed on this platform. They use this adenovirus. So adenovirus, everyone, I think there are so many vaccines who already use adenovirus. So what is famous, what is different between, what is the difference between adenovirus that is found in the nature and the adenovirus that is used for a vaccine? So this adenovirus, it's a flu-causing virus in chimpanzees, monkeys, which are relatives of monkeys, chimpanzees. So this virus, it causes flu in chimpanzees. That's why the first two letters, chimpanzee, and then adenovirus in the vector design, what they call is CHADOX and OX for Oxford. So Oxford scientists, they mutated this adenovirus in such a way that once it's injected into a human body, it doesn't replicate. And if the virus doesn't replicate, it's safe. That's simple logic, right? So that was the platform technology adenovirus that Oxford scientists had developed. And luckily, they had trialed this platform technology already in development of other vaccines, like vaccine for MERS, SARS. So they already knew that this platform technology is safe in human. They did not know the vaccine will be effective, but at least they knew this platform technology will be safe. So merely what they had to do is take out the MERS virus, MERS, which is Middle Eastern Respiratory Syndrome, and just what we think is COVID-19 originated from bats. MERS is a zoonotic disease which gets transmitted from camels, and is because it's Middle Eastern, because it's more, camels are mostly used in Middle East, so that's why it's called MERS. So the simple thing, once the sequence was published by the Chinese scientist in January, Oxford scientists quickly went to work. Once they had the sequence of the whole sequence, genome sequence of the virus, they started quickly doing their work. And what they simply did was change the virus inside. So put the DNA of the coronavirus into this adenovirus, okay? And when you have this adenovirus, when you have your DNA into the vector, so upon, suppose Oxford AstraZeneca vaccine is given intramuscular. So once it comes into the contact with human body cells, this adenovirus, it enters into the human body, enters into the human cells. And once it enters the human cells, it releases this DNA into our human cell, once it into a single nucleus. And as we all know the biochemistry, the DNA then breaks down to mRNA, and mRNA is translates the protein for the spike protein. So that's how the coronavirus mRNA enters the human body. And now the human body, because it has now got the mRNA, it can now start synthesizing the spike protein of coronavirus in our body. So once it's inside the cell, and the cells start producing and the spike protein. The spike protein is then displayed on cell surface receptors on top of the cell surface. So when the cell is circulating in the body through the bloodstream, this spike protein stands exposed. The other way that will generate an immune response is when the cell dies. So even after the cell dies, these are proteins, right? So these proteins are still stable in the body and they are still circulating. So when these spike proteins are circulating, there is something called, in immunology, called antigen presenting cells. These are mostly dendritic cells. Those who are not familiar with antigen, what antigen presenting cells are, let me put in simple terms that these are our police of human body. They are constantly scanning and searching for any foreign intruder. Like, they know what should be in our body and what shouldn't be in our body. So when these dendritic cells are constantly like act as police, and then they encounter the spike protein, and this is okay, the spike protein doesn't belong to my body. What's it doing here? So it goes and captures it, and then it asks for more help, like more police force comes in, and then it gives it to a helper cell, like listen, we have found this foreign protein in our body, we should do something about it. So then the helper T cells, which are also called the CD4 T cells, they come into action and they interact with the antigen presenting cells. So what do the helper T cells do? The helper T cells goes and alarms other immune cells in the body, such as B cells, and B cells are ones that produce antibodies. So it goes and alerts B cells, like, listen, this is a foreign antigen that is circulating in our body. We have to eliminate this. Can you please start producing antibodies for this? And there you go. Our B cells start producing antibodies. And the other way is the B cells also then the vaccinated cell, which is already exposing the spike proteins on its surface, that also comes in contact with the B cell. It binds with the antibodies, and voila, the spread of the virus stops in the body. So the very simple concept, like how the immune cells get activated. We talk about generating antibodies and everything, but which are the actual cells that do it? So these are the cells that are actually important, and it's a very, very intelligent system. Our body is more intelligent than we are. It's such a wonderful system that it works within, and we don't even realize what's happening in our body. So that's how, in short, the vaccine works. So when they encounter the virus, The antibodies, they will go and bind on the spike protein, and then the spike protein won't be able to replicate, it won't be able to enter into any other human cells, and thus it stops the spread of the virus in our body. So a very simple concept that these antibodies work, and most of the antibodies, most of the vaccines are based on this concept. Until recently, the mRNA vaccines came into picture. So I'll start now with the paper discussion. So this was the paper that came in. This is not the first paper in Lancet. So they published phase 1, phase 2, and phase 3 trials as well. And so this is the paper that was published by Oxford in December 2020. They had already released the data in the media in November or something earlier, but it was still some time for the. Sorry, can someone please mute the microphone? So I just want to highlight one thing over here is the first author of this paper. The first author of this paper is a biostatistician. There are many big names, many big scientists in this paper, but it's just, I thought it's worth mentioning that the first author of such an important paper is a biostatistician, because there is tons and tons of data that has to be analyzed. After the science part is I think when it's done, it's okay. But to put out this data to the world and make sure that data is collected and analyzed with integrity is the job of a biostatistician. And that's why it's one of the important discipline in our field as well. Like biostatistics is so important. So I'll just now quickly move on to the paper. So how was the trial design, the Oxford AstraZeneca trial design? So it was randomized, blinded, multi-center trial. So those who are not familiar with these terms, I will just quickly explain what these terms are. Randomized. Randomized means when you enroll yourself into a clinical trial, when you participate in a clinical trial, you don't know whether you will be in a control group or whether you will be in the treatment group. So that's randomization. No one knows. Even the principal investigator won't know, and even the personnel working on the clinical trials, they won't know which arm you will go. It's a computer generated system these days, and it's mostly the computer acts is like every participant goes into 1:1. So one control, one treatment, one control, one treatment, but no one knows who will go in which group. Blinded. What do you mean by blinded? So the person giving the vaccine to you, he doesn't know if it's a control or an actual vaccine. It can be a saline, it can be a vaccine. So that's blinded. And even you don't know, person giving the vaccine doesn't know. It can be unblinded if there is what we talked about, serious adverse reactions. If there is a serious adverse reaction, then go and they unblind. Means obviously they note down the catalog number and the batch number and everything. If they want to find out where this vaccine was, the person on the treatment group or the control group, they can go and find it out. That's called unblinding. But until then, if there is no serious adverse reaction, they don't know. It's only at the end of the trial, at the end of the study, they will unblind all the patients. And multi-center is obviously self-explanatory, that it's not carried only at one clinical facility. It's carried out at multiple clinical facilities. And in this case, it was multi-center trial in terms of, it was a global trial. So the trial sites were in UK, Brazil, and in South Africa. 2 sites in UK, 1 in Brazil, 1 in South Africa. Participants that were enrolled in these studies, they were all aged 18 or over. As I previously said, all the participants, they were assigned to groups in 1:1 ratio, just to make sure there's no there's no more participants in one particular group as compared to another one. 2 doses were given at the interval of 28 days. So there was this one controversy, you might have read it in the news. So they made a mistake during the trial. So for a small subset of patients, they decided on a dose, and everywhere they gave the same dose, which was 500,000 virus particles. Okay, but in small subset of patients, around 4,000 of patients, they did a dose calculation mistake, and I will explain what the dose calculation was. So they of the first dose they received was a half dose. So they received 200,000 virus particles. When I say 200,000, it's 2 lakhs. Well, like we use lakhs in India. So 2 lakh virus particle in the first dose, and then they realized, oh, we made a mistake in our dose calculation. So they wrote to the MHRA, the regulatory authority, the UK Medical Health and Regulatory Authority for the UK, and they said, like, listen, we have made this mistake, but we have just given a half dose to the first. So what should we do? So the MHRA suggested them, okay, the second dose, you give a full dose. And they were very open. They mentioned it in the paper as well. It's nothing controversial. It's the scientists later on raised questions about it. But I will explain you why this controversy happened. And how did the monitor after getting the patients, if even after one dose, is like, they were continuously taking their nasal swab testing, and like, there was monthly follow-up just to make sure, are they getting infected, the fevers, overall conditions were monitored after getting the vaccine. In total, they recruited around 24,000 participants globally. But in the Lancet paper, when they published the data, they had published it for 12,000, because already Pfizer had published the data, and it almost was like a race. So they quickly hurried it, I think so, and they published it for 12,000 patients. But in the phase 3 trials, there were total 24,000 patients. So coming back to the dose calculation, what mistake did they did? So when they weren't open, when they initially released the press statement, they didn't clear. They said, oh, they made it look like, oh, we gave 1/2 dose and we gave another full dose. And it was only later on that was brought to by attention by other scientists globally, in US and in Europe. It's like, no, like, you did a mistake, and like, why you're hiding it? And they were not hiding it. It was there in the paper. But then there was like, once you start hiding things in research, and especially research like this, like, when the entire world is waiting for a vaccine and you want to know whether it's safe and effective, there were questions raised about the vaccines there were doubts raised, and it was pretty dampening for all the scientists who were working on these vaccines. But what was the mistake? So the university had a contract manufacturer, which is called a company near in Oxford Biomedica. So they were manufacturing the vaccines for the clinical trial, and there was another company in Italy called Advent. They were manufacturing the vaccines as well. So the company in Italy, for the method for dose quantification, they were using qPCR, and if you know, qPCR is qualitative polymerase chain reaction. And the manufacturer in UK, they were using spectrophotometer to quantify the dose. Obviously, 2 different methods, totally different results. In the end, it was found that qPCR was more effective, but because majority of the patients had received vaccines whose dose was calculated by spectrophotometer, they continued with using the spectrophotometer to quantify the dose. But that's what happened. Nothing wrong. All the way doing their work with integrity. But that's how, like, science is. That's how research works. To save Oxford from all this controversy, the best thing was to eliminate the low dose group from analysis. But then the sample size would have been reduced significantly. From 12,000, you're already taking out 4,000 people out. So they would have had to spend another months recruiting those patients, and it would have just delayed. And like, it was just vaccine nationalism in the end. Whose vaccine will first hit the market, whether it'll be a US vaccine, whether it will be a British vaccine. So I think they just hurried up. The moment Pfizer data came out, I think within weeks, they just published that data. So in the hindsight, they could have just taken out the low dose group, but it's okay. It all worked out. So what are the results? Result is safe and efficacious. Yes, it's absolutely. And I'll just go to another controversy or not the controversy, but there was a certain side effect that came into a highlight after the vaccine went into market, and then more people started taking it. And why this side effect didn't pop up in clinical trials, I will explain that as well. So overall, the vaccine was found to be safe and efficacious. 2 full doses, 62% efficacy. So for WHO to approve a vaccine, you need an efficacy of 50%. 62%, well and good. It was still, Pfizer and Moderna results weren't out. Oh, Pfizer is 95%, Oxford vaccine is 62%. Not so good. But first, this was an interesting result. And those 4,000 people who received first half dose and second full dose, 90% efficacy. It's really good, just like Pfizer vaccine. But the scientists dismissed these results. There's like very few participants. At I'm seeing 4,000. I think it was around 3,700 or something. We can't be confident at 3,007 participants whether it's actually 90% or not. So those were, they said, okay. And then when it went to the UK MHRA, and UK MHRA said, like, then what's the dose that should be actually given? Like, should we go first half when we will actually start vaccinating the wider population? So what dose which should we actually do? MHRA said, you know what, you admit it was a mistake. So, and because the trial, the number of participants was so few, we can't trust the low dose and the high dose data. You have significant population in the high dose arm, 2 full doses. So we'll go with 2 full doses, 62% efficacy. Okay, that's the dose then, 500,000, which is 5 lakh virus particles. That's the doses they went. Then further trials were conducted. Even then the market, the vaccines hit the market by March or something. I think vaccination had already started. There were more trials conducted in the US. And the US trial showed just after 1 dose, just after the first dose, the Oxford AstraZeneca vaccine offered 76% protection, which was much more higher than the 62% efficacy, which was good news. You know, just first dose can stop the spread of the pandemic. So 70% protection, yes, really good. Scientists were very pleased. Then came another news that, you know, instead of 28 days gap, we can increase the gap from 28 days, 4 weeks, to 12 weeks. Why? They did another study. 12-week gap actually further increased the protection offered. 12-week gap went to 82% protection, which was then, even in UK, when they started the vaccination, it was 4 weeks. And then when this clinical trial data came out that 12 weeks interval offers higher protection, they changed it, and it's currently at 12 weeks. And there is also explanation for why, if you guys are interested, why the low dose and the high dose had higher efficacy than 2 high dose. There is a really good scientific explanation. It's very immunology based. It's very complicated, but those who are interested, I would go and suggest and read about it. There are a lot of clarification that came on by a leading immunologist in the field, and they said, you know what, this data is not actually wrong. The 90% efficacy might be true. So those who are interested can just have a Google search and read about it. The numbers just started getting better and better for Oxford vaccine. When the real world data came out, when I say real world data, when there was mass immunization going on, like millions of people were getting vaccinated in UK and in Europe, the vaccine was actually up to 90% efficacy. This was phenomenal efficacy. Just forget about the efficacy. What the vaccine actually does is 100% protections against severe disease and hospitalization and death. This is stunning. And people, when I speak to my family and friends back home in India, and they think like, people are, oh, you know what, I had my 2 jabs, and I still got infected. Why? I said, like, yeah, there is still a 20% chance that you will get COVID. What the vaccination does is saves you from severe disease. It saves you from hospitalization. It saves you from dying. And there are so many, there are so many examples, like, person with no comorbidities at all, just because he wasn't vaccinated, he died with the Delta variant, the variant that's spreading now in India and across UK as well. But person who had 2 full doses in the same family, that person survived. Why? Same age group, everything same, because they had 2 doses. They recovered at home, but the person without vaccine had to be admitted in hospital for more than 6 weeks. And now, even after 6 weeks of treatment, that person still goes on to die. And the person who had 2 full doses of vaccine, he got COVID, he got ill, but he recovered. That's the beauty of vaccination. So we should, we should always try and educate those who are not from the science background. What are the advantage of vaccine? This is the advantage of vaccine. All right. Let's come to the side effects, which was again highlighted quite a bit. Yeah. Oh, there is blood clot. There is this Oxford AstraZeneca vaccine causes blood clot. There was a whole meta-analysis of the data that was done, and UK MHRA and European Medical Agency, they came out and gave a statement. Yes, that we think there is a causal link, but the benefits outweigh the risk. And how much they outweigh? This is the stunning figure. Look at this figure. 27 cases of blood clot in 2 crore people who were vaccinated. These 27, like, more people get blood clots without vaccination than those 27 who got. But these days we get more excited by the negative news than the positive news. This figure wasn't there. And the European Medical Agency and UK MHRA had to come out and say the incidence is so low. And out of those 27, I would, if I am not wrong, I think only 3 or 4 people died. And let me tell you, these 27 people, they do have some other medical conditions associated with it. So this is the blood clot risk they are saying is such minuscule, is so minute, that it shouldn't turn off people from vaccination. 27 cases in 2 crore people. I think we should make the decision for ourselves. Is it really a risk, or is it really a benefit? I clearly said there is no risk at all, honestly. There is a causal link the scientists have put out. It's mostly because of the vector, but there are always some side effects with the vaccine. So just wanted to put out this out about the side effect. Once like to acknowledge the scientists who were involved in this, Professor Sarah Gilbert, Professor Adrian Hill, and Professor Andrew Pollard. The reason I thought it's important to put out their names, and you must have heard them in various media interviews, is this was developed at a record pace, like 8 months or so. They had published the phase 3 finding. There are reasons why they did it in 8 months. There were no red carpets. There were no red tapes. Sorry, there were no red tapes. All the bureaucracy was put aside. They got all the clearances from the government, which normally take 2-3 years. They got in weeks. They had already a platform technology which was very safe, and they just went on it. And it's commendable. Like, these are not only the 3 scientists. There were a whole team working on this. The whole Jenner Institute, which worked on this. And also later on, AstraZeneca with its private funding that came into picture. And the reason I put out the picture is, yes, it's academic research, but they commercialized this research. How? So as we all know, the Oxford vaccine is now called Oxford AstraZeneca vaccine. AstraZeneca is a British-Swedish pharmaceutical company headquartered in UK. And obviously they collaborated with AstraZeneca, just like the German company Pfizer, BioNTech collaborated with Pfizer because if BioNTech is a small company, they didn't have the resources, they didn't have the money to conduct these large-scale clinical trials, they didn't have the logistics behind those clinical trials. Same with Oxford, it's a university minded. So they had to collaborate with some big company, and why not a British company, which is headquartered in UK. So they collaborated with AstraZeneca for funding reason, obviously, because running clinical trials is huge. The cost is like billions and billions of pounds. So that's one reason. And the best thing, I'm pretty sure this doesn't apply to the Pfizer vaccine or other vaccines that are being produced, but Oxford and AstraZeneca, they're currently selling their vaccines for no profit until the pandemic is there. That's again commendable. I think that's really good. And not the science part of it, but Oxford AstraZeneca vaccine, it cost them around 400 rupees, or like if I'm talking about rupees, or in dollars is like $4. And Pfizer Moderna vaccine is like $40. They're 10 times more expensive. And just, I showed you the data, they are equally effective. The technology is different, but it's almost equally effective. 10 times more expensive vaccine, but equally effective. Vaccitech. So this is how Oxford University commercializes its research. So this is a spin-off company from the Oxford University, which is co-founded by Professor Gilbert and Professor Hill. And this is one thing, like, it's encouraged a lot. I just on purpose, I put on this slide, not that is related to my talk, but how it's important to show that how academic research can be commercialized. This company is now, from nothing, this company is now valued at more than $100 million. It's listed at New York Stock Exchange now. Professor Gilbert, who are actually university staff, they partly co-own the company. They are co-founder of the company. The Oxford University is one of the majority stakeholder in the company, and it's an excellent source of revenue for the company now and for the university, because that's how you put in effort, you put in money. It's years and years of research. So this vaccine must came out in 8-10 months. But to develop the technology platform, the chimpanzee adenovirus Oxford One, the technology platform, they own the proprietary rights, they own the intellectual property rights of the Vaccitech company. This took them years of research. And currently, the vaccine is sold for no profit, but in the future, obviously, they will make some money. But until the pandemic is over, they won't be making any profit, which is again commendable. So that's how you commercialize your academic research. Current studies. So this is unfortunately a very short part of my talk. What is Oxford doing now with the vaccine? They want to administer the vaccine as a nasal spray. This clinical trial, it's in phase 1, and they are now currently working on it. They have already started recruiting volunteers, and they want to use it as a nasal spray. And what's the benefit of using nasal spray? So deliver vaccine directly to the primary site of infection. That's the main. They do feel that, when you deliver, the immune response is much better because we all know if we eat the virus, it won't cause, I don't think it will cause any, because our pH in our gastrointestinal system will just destroy the virus. So that's why covering the face mask is important, because it enters through our respiratory system. So if there is a strong immune response at the site of infection, maybe the scientists think it will be better in controlling this virus. And the other thing is, it may increase uptake. Some people are just not taking virus just purely because they have needle phobia. So, which I think is a really good reason to go to the nasal route by nasal spray. So let's see how these results go, whether they do. There are actually quite a few vaccines, the flu jab and everything. They are already given through nasal spray. So I don't think why this won't be less effective. Cool. So current treatment, what works and what does not? Dexamethasone, tocilizumab. Tocilizumab, as you know, it is worked in treatment of osteoarthritis. It is also used in some cases for cancer patients as well. It's a monoclonal antibody. So these two, and dexamethasone, we all know it's a very cheap steroid. And again, this study was done by Oxford. And dexamethasone is working wonders in controlling the severity of the disease. So what's the problem? Excessive use. Excessive use is the problem. It's such a cheap. I think in India, maybe you get a strip for 4 or 5 rupees. That's how cheap it is. But excessive use of steroids, that's what causing the problem. And that's what we need to, we don't practice medicine as such. The doctors need to focus on, like how much dose you're gonna give of dexamethasone you're gonna give, because what does COVID does? It causes a cytokine storm. Yeah, like, a lot of cytokines secreting in the body. What does dexamethasone does? It suppresses the immune system, because yes, you need an immune response, but when there is too much immune response, your immune system starts attacking your own body. So that's where dexamethasone was so useful. But you have to be careful with what dosing that. It's still widely used in UK. There's another drug, and I was in so many WhatsApp group constantly from India, and like, oh, who's got remdesivir? Can someone ask for remdesivir? No one is getting remdesivir. But a trial that was concluded, it showed like remdesivir is of little or no use. And I spoke to my doctor friends in UK, like, do you use remdesivir over here? He said no. I said no, like maybe in very rare cases when there is absolutely nothing working, they go for remdesivir. But it's not on the line of treatment for COVID, for management of COVID. And I think this something again, like, and it's just not one trial. There are quite a few clinical trials, and you can just go and you can just Google for WHO solidarity trial, and there were another few studies done in US as well. And they concluded remdesivir is maybe a little benefit, but the cost doesn't approve of the benefit that is. So this is something to again think of. There's another development that is being done is molnupiravir, which is developed by Merck. Sorry, so apologies for the spelling mistake over here. It's Merck. It's developed by US company Merck. It's in phase 3 trials now. And hopefully there are a lot of hopes on this particular medicine, because yes, you have a vaccine, but you also need, people are still getting infected, some age groups might still get infected, and you need something that is targeted. Why? Because all these treatments now, this is the latest headline from our government of India, the new guidance. All these hydrochloroquine, they all with ivermectin, and now people like talking, I don't know, methylene blue or something. It's all. There is nothing unless and until anything is backed by scientific data, unless and until some clinical trials have been done. I don't know how much to trust these other medicines. So let's be careful. But the thing is, like, we don't, as pharmacists, we don't practice medicines ourselves, but the doctors, they have to think about it and think and just read more literature, what's happening, what trials are done. There are thousands of clinical trials for treatments going on. And I just wanted to shed a light on what actually works and what doesn't work on management of COVID. Way out of pandemic. So how do we come out of pandemic? Will we ever have a post-COVID era? Yes, obviously, pure logic, common sense, full vaccination, 2 full doses, that will save us from that's the way that we will come out of the pandemic. But what now the government is doing in UK is full vaccination, plus they're already planning for a third dose, a booster jabs in autumn, every 6 months. So it's not just 2 doses. Even after 2 vaccination, people will get vaccinated every 6 months or something like, every year. because now the scientists believe that coronavirus is there to stay with us just like a common cold flu. And the problem is why booster jabs every 6 months? And that's the problem with COVID vaccines, not just Oxford AstraZeneca, but also with Pfizer, Moderna vaccines as well, is the level of antibodies that are generated after first 2 doses, they start going down after 6 months. So you don't have the same level of protection. In some patients who are infected with COVID, okay, they said, oh, you've got a natural immunity. That's what happens with other diseases like smallpox or measles or mumps. You get infected once and then you don't get it later on in life because you have developed a memory. Your body has developed a memory and you won't get infected again. But the problem with coronaviruses or the antibodies that are generated following infection or following vaccination is the level of antibodies goes down. And this data is something very under the carpet, but in some of the patients, even after the first dose, in some patients there were no antibodies detected for some of the patients that were vaccinated. So it's kind of scary. So because the level of antibodies do drop down, in some people they can stay longer than 6 months, in some people in 6 months they drop. There is an immune response, but it's not a prolonged immune response. So full vaccination plus booster jabs every 6 months, that's one way. Entire world needs to be vaccinated. And I will give an example of UK. It almost looks like it came out of pandemic in October, November last year. Then there was a variant in UK, the Kent variant that we know. Now I think it's called Alpha variant or something. So it gave rise to second wave in UK. Then it came down. Almost everything was going to open as normally. We were in lockdown for a good 3-4 months from November. We thought everything is gonna be open. And now we have the Delta variant, which originated in India. Obviously, there was still global travel going on, and now the cases in UK have started to go up again, and it's 75% of the new cases are the Delta variant in UK. So, how long you going to close the borders? That's not the solution. So the entire world needs to be vaccinated. UK has a phenomenal record in vaccinating people. I think more than 50% of the adult population have received the full dose, 2 doses. But unless and until the entire world gets vaccinated, there is no way out of this pandemic. Yes, vaccination protects you, but once you get the disease, you need some targeted treatment. All the medicines that we have, dexamethasone, tocilizumab, or remdesivir. Remdesivir was developed for Ebola. It's a broad spectrum antiviral. All these treatments are repurposed medicines. They weren't developed for targeting COVID. They were developed for targeting something else. So you need something, and that's why there are a lot of hopes pipped on molnupiravir by Merck, because you need some targeted treatment just for that particular virus. How to tackle mutations? The Delta variant, there are some preliminary data coming out that the Delta variant, the vaccines, they are only 60 to 70% effective against the Delta variant. So the worst has happened actually. The response of the vaccine, it has actually gone down. So it takes time, because to tweak the okay, you found a variant, you need a different spike protein and those things. You need to tweak the vaccine. The vaccine tweaking takes time. It's easily 3-4 months of work to maybe start producing new vaccines. So how to tackle mutations? Now the scientists are spending the time in predicting mutations. Can we predict how the virus will mutate in future? And can we already have the vaccines in stock produced, mass produced? And once this is the mutation, we have the vaccine ready. Let's start vaccinating people with this. Can the mutations be predicted? Because there has to be a strategy to tackle mutation. Otherwise, it will just keep getting worse and worse. And I will tell you why. I just mentioned shutting borders and lockdowns is not the way out of this pandemic. Let's hope on the brighter side. Let's hope COVID-19 becomes less virulent and it circulates like a common flu, just like we get cold, common cold, just we like get cough every once a season. And it's not life-threatening. Your body recovers. You take some paracetamol, you take some ibuprofen, you just rest for a day and you feel fine. Can COVID-19 becomes less virulent? I mean, when I say less virulent, is will it spread less and will it cause less severe disease? That's what I mean by less virulent. Has the COVID-19, will COVID-19 become less virulent and circulate? If that happens, nothing like, oh, then we don't have to take, maybe like, just like we don't have something called flu jabs in India, but here we do, like, once every autumn, we just go, we take a jab and it just protects us against the common cold, like the influenza virus, just like a flu virus. And we need a lot of luck. Why I say a lot of luck, and this is the important bit, luck. If the virus mutates and becomes more aggressive, that's, this is bad. If this happens, this will be devastating. All the work that is done by countries all over the world, this time and effort they have spent, the lockdowns, the hardship that people have went through since last 1 year, students have went through, it will all be undone. If the virus mutates for the worse, it becomes more aggressive and the vaccines don't start working at all. That's a scary situation. That's why I say we need a lot of luck for a way out of this pandemic. Okay. Post-COVID era, will, when, why, how will a pandemic ever again hit us? Let's analyze this. Pandemics are no more once in a century events. Okay. The last pandemic that hit the world was in 1919, the Spanish flu. The World War was going on. There were a lot of troops movement going on from US to Europe. Within Europe, the troops were moving and the Spanish flu spent like anything. And it killed millions and millions of people. Since then, we don't remember having a pandemic. There was an endemic with SARS and everything. We didn't have a pandemic. So after Spanish flu in 1919, just look what we got in last 15 years. SARS, MERS, Zika, Ebola, Nipah. Just in last 15 years. They were at endemic stage, but these viruses, they are still circulating among humans. Remember that. And these viruses, they might again mutate for the worse. It is not that we have totally got over that. There are still cases of MERS. SARS, we have controlled it. Ebola, there is still some cases in Africa. So these viruses, they might mutate for the worse. And, we might have a next Ebola pandemic or a next Zika pandemic. We don't know. And 70% of the disease, not just these viruses, but which causes pandemic, just like including HIV, they are of animal origin. They don't come from us. They are zoonotic disease. When they get transmitted, when they overcome the species barrier, that's when these different diseases start arriving. And because our human system is not ready for this, because there is a species barrier, we can't fight this. And that's the reason they become pandemic. Someone asked how many coronavirus, I was listening to the talk before and someone asked, how many coronaviruses do you think there are in the nature? Look at this figure, 900,000. 900,000 is 9 lakh coronavirus. It's an estimation. I think there is 1.5 million, which means I say they are in the nature, in animal. It's estimated. This is a very generous estimate. And we are just waiting, just like from bats or from camels, we have got previous pandemic. We are just waiting, like, next pandemic might come from a mouse or a cat. We don't know. It's just a matter of time when this coronavirus will cross that species barrier. And what's happening? Why does it happen? It's just increased human-animal contact due to we are just not taking care of our nature. It disturbs the habitat, and because there is increased human-animal nature or contact, these animal viruses, these zoonotic viruses, they are coming into human contact. How? So the theory behind the SARS virus is in China or in Thailand. I'm sorry, I don't actually recollect where it actually started, but I can tell you the story. There was deforestation that was going on, and the deforestation which was happening in that area was a rich habitat for bats, and bats, they were dying out of this deforestation because bats lives on trees, right? So they were dying out of this deforestation, and when some bats were ill, and when these bats were dying, farm pigs, when they are on the farm, they were coming into contact with the bloods of these infected bats. And so the pigs, they come in contact with the infected bats. If it is a farm pig, people in other countries, they eat pork, which is pig. So then this pig, which is infected with the bat blood, which had some virus, then it does cross into the pig, and then some people, it goes into the restaurant, the chef and everyone who is handling the pig, which is not yet cooked, so the virus is still thriving and multiplying inside the pig. They touch the pig and they have the virus on their hand, and then it starts from one person. There is always a zero patient where it starts, and then it starts spreading. So increased animal human contact due to deforestation, it disturbs the habitat, and that's how pandemic starts. Global travel. Imagine if there was first case was identified in China for coronavirus in Wuhan. What if there was no global travel? It was never, but that we cannot stop. Even in the pandemic, people were traveling across countries, important travel, but still people were traveling. Eating habits. Eating food which is of animal or bird origin, suppose say chicken, not just animal I'm talking about, but any kind of birds, which is chicken, or animals, I'll just give you an example of wet food for market on China, which scientists believe these are the breeding ground for pandemics, and the images were too ghastly for me to put on this presentation. I mean, you can't bear the sight of these wet food markets. Each and every animal you ask for, which is there on the planet, you will find in these wet food markets, and they are there in close contact with each other, all the animals, and they are sold right and there itself. They are live and they are butchered over there, they are killed over there, and then their meat is sold. There is blood everywhere. It's all mixing everywhere, and that's the breeding ground for the pandemics. Eating habits, people want to eat this meat, but then they bring the viruses into the human circulation, and then there is more overcrowding. More people from villages, they are living into the cities, overcrowding. And obviously for pandemics, when there is overcrowding, there is rapid spreads. Very simple solution: vaccines, everything. Yeah, you can prepare for it. I think the only way out of this pandemic is preserving our nature, preserving the biodiversity is the only way to avoid future pandemic. But you talk to any other person, when will another pandemic hit us? Talk to an immunologist, talk to people who study pandemics. They will say maybe in another 10 years there will be another pandemic. So which is a very scary situation, but they will become more often, considering with the number of endemics that we had, like SARS, MERS, Ebola, Zika, Nipah. The list is long. Swine flu. So I think the next pandemic will hit us very soon. How can we avoid it? By preserving our nature, by preserving the biodiversity, reducing the animal-human contact. That's the way out of these pandemics in future. So I thank you very much. Thank you for listening. Hope I didn't go over time. Okay, thank you very much. Thank you for listening.
Thank you. Thank you very much, sir, for your valuable words. Now moving toward the question answer session, some participants have put the question in the chat box. So I can ask you. Can I ask you question?
Yeah, sure. Please. Yeah, I'm happy to take any questions. Yeah.
Yes. So first question we have got, that is what is the role of immunosuppressant in the COVID-19?
So COVID-19, when people get infected, what I use the term as a cytokine storm. What are the cytokines? Interferon gamma, TNF alpha, interleukin 2. So our body responds to this and they start producing these cytokines in much more amount sometimes than it's where it's needed. And then it causes inflammation in the lung, not just on the lung, some other organs as well. It affects kidneys, it affects livers. And there is this overt production of this cytokines, of this overreaction of the immune system that causes most of the people severe illness. And to control that, that's why we need immunosuppressants.
Okay. The second question, as the storage temperature of the Pfizer and Bharat Biotech vaccine is in negative, so how it stay working in our human body?
Those vaccines, once I showed a slide, right? So once they come into the human, once we are vaccinated, and once the mRNA or the DNA, once it gets in contact with human cells, then they're fine. It's just a protein. It gets degraded. And we have got mRNA, DNA everywhere in the body, right? It's stable. That's how we are created. So once it's there in the body, the mRNA is there, and it starts translating for the spike protein. So, yeah.
So next question, which one is better between the Covishield and the Covaxin? People very eagerly wanted to know that.
Yeah, I think both are good. You know what I will say? Covaxin, Sputnik, Covishield, you get what you get it now.
Yes.
Whatever vaccine is available now, you get it. It's not a competition. You don't compare vaccine. I was speaking to my friend, and I think he is in the age group in India. He is in the age group where he can get vaccinated. And he's like, why haven't you got vaccinated? I'm waiting for Pfizer to come to India.
Yeah.
This is not a competition, guys. Remember this, right? Your first thing is get vaccinated. You will have some sort of protection. So get any vaccine that you get. This is not a race. This is not a competition.
Okay. The next question have asked that this question answer you are already given. So I will ask you the next question. Why Covaxin is not yet approved by the WHO?
It's approved for emergency use in India, but they haven't published their phase 3 results yet. They have to show their phase 3 results. It was just because the pandemic was raging, they thought it's effective. But if you want it approved by the WHO, there is a panel of scientists who will assess its safety and efficacy. They are not trusting. I think Bharat Biotech did a brilliant job in developing that vaccine, but they have to publish the phase 3 results for WHO to be convinced that it's a good vaccine.
Okay. Next question. Why WHO removed the remdesivir from in the corona treatment?
Yes, I think there is. I think, and everyone should do it, right? Just Google a solidarity trial by WHO, and it's quite striking. The drug which was touted as just like hydroxychloroquine in the beginning. Oh, it will. It's so important for treating COVID. Same thing happened. It was just the hype, but when the actual data started coming out, it's a broad spectrum antiviral. It's not it's what I said was we need something like targeted treatment targeting this COVID-19 vaccine, COVID-19 virus. So it's not targeted. Remdesivir was developed to treat Ebola virus. Let's keep that in mind. It's not specific for COVID-19. And the clinical trial data showed it's of little or no use. So WHO said, like, people were grappling for they said, listen, it's not that effective. They have actually removed it from official line of treatment. It's a big step, but I think some doctors do believe it's still good. I don't know. I'm not a medic person, honestly. We are just like biomedical scientists. So it's for the doctors to make these decisions whether they should still give like drugs like remdesivir, hydroxychloroquine, ivermectin, which like the WHO clearly feels they're not any better in managing COVID.
Okay, so you answered all of the questions. So would you like to give any message to our people about the safety of vaccine?
Yes, I think it's very plain and clear. And in the slide that I showed, way out of pandemic, it is the only way. Otherwise, we will be in the lockdown and people will be going through hardships for another year, another 2 years. This isn't good. We'll have coronavirus cases coming in place like India for another year or so, unless and until people get vaccinated. There is no way out of this pandemic. All vaccines, they are safe. Okay. Some people might have some side reaction, which is understandable. But again, the benefits of the vaccine, they are for the better good. The benefits far, far more outweigh the risk involved. One click, simple message. Whatever vaccine you get, Covishield, Covaxin, Sputnik, you get vaccinated. That's how you're gonna come out of this pandemic. Yeah, I think all the vaccines are safe. Just take whatever you get. I think you just get it. All vaccines are safe.
Yes, yes.
And that, as pharmacists, I think we have to send this message loud and clear in the community. There was not believe those WhatsApp forwards that we get. And it's our job as healthcare professional, as pharmacists, to educate and go out and tell the people strong and loud, get vaccinated. And if anyone is talking against vaccines, let's bring them down. Let's bring the anti-vaccine campaign down. Like, that's not going to do any good to the humankind.
So definitely, as being a pharmacist, is our duty to guide or tell the people about that vaccine use and the safety of vaccine. So thank you so much, Dr. Jagdish Mahale sir, for accepting our invitation as a guest speaker within a very short period of time and sharing your valuable knowledge with us. Thank you very much, sir.
Thank you very much. Thank you for inviting me, Gopal. And thank you very much. It was nice giving this talk. Thank you so much.
Yes, thank you. So now all the participants are requested to fill the feedback form, which will be shared shortly on the chat box and the WhatsApp groups. So tomorrow, all participants are requested to join the second day of this international e-conference at 10 AM on our college YouTube channel. So here I declare with the prior permission of our principal that today day 1 session is completed. So thank you all. Thank you very much.